One of the most important biological mechanisms through which alcohol increases cancer risk involves the formation of a toxic compound called acetaldehyde. When alcohol is consumed, the body immediately begins the process of breaking it down so it can be removed from the system. The active chemical in alcoholic beverages is ethanol, and once ethanol enters the bloodstream, it is transported primarily to the liver, the organ responsible for metabolizing most substances that enter the body. Inside liver cells, specialized enzymes begin converting ethanol into another compound called acetaldehyde. This step is necessary because ethanol itself cannot be eliminated efficiently until it is transformed into other molecules. However, the intermediate substance created during this process, acetaldehyde, is far more harmful than ethanol itself. Acetaldehyde is classified as a carcinogenic substance because of how aggressively it interacts with biological molecules. Once formed, it can bind directly to DNA and proteins inside cells. DNA carries the instructions that regulate how cells grow, divide, and repair themselves. When acetaldehyde attaches to DNA, it can distort the structure of the genetic code. This distortion interferes with the normal replication process that occurs when cells divide. During replication, the cell must copy its DNA precisely so that the new cell receives an accurate genetic blueprint. If acetaldehyde has altered the structure of the DNA, the copying process becomes more prone to errors. These errors can result in mutations, permanent changes in the genetic sequence. Mutations themselves are not always harmful because the body has several layers of repair systems that constantly monitor and correct DNA damage. However, acetaldehyde also interferes with the very enzymes responsible for these repair processes. When DNA repair mechanisms are disrupted, mutations that would normally be corrected may remain embedded in the genetic material of the cell. Over time, the accumulation of these mutations can affect genes that control cell growth and division. Some of these genes function as brakes that prevent uncontrolled cell proliferation, while others regulate when cells should die and be replaced. If mutations disable these protective systems, cells can begin dividing in an uncontrolled manner, which is a central characteristic of cancer development. Another important factor is the level and duration of acetaldehyde exposure. The body contains another enzyme called aldehyde dehydrogenase that converts acetaldehyde into a less harmful substance known as acetate. Acetate can then be further broken down into carbon dioxide and water, which the body can easily eliminate. Under conditions of occasional alcohol consumption, this detoxification pathway can process acetaldehyde relatively efficiently. However, when alcohol intake is frequent or excessive, acetaldehyde may accumulate faster than the body can neutralize it. This prolonged exposure increases the amount of time that cells and tissues remain in contact with this carcinogenic compound. Certain individuals are particularly vulnerable to acetaldehyde accumulation because of genetic variations in the enzymes that metabolize alcohol. In some populations, the enzyme responsible for converting acetaldehyde into acetate functions less effectively. As a result, acetaldehyde remains in the body for longer periods, intensifying its damaging effects on cells. This variation demonstrates how the risk associated with alcohol metabolism can differ between individuals depending on how efficiently their bodies process this toxic intermediate compound. Acetaldehyde does not only affect the liver. Although the liver handles most alcohol metabolism, acetaldehyde can circulate through the bloodstream and affect many different tissues throughout the body. Cells in the mouth, throat, esophagus, stomach, and intestines may all be exposed to this compound. In these tissues, acetaldehyde can directly damage DNA in the cells lining the digestive tract, increasing the likelihood that mutations will accumulate over time. Because these tissues are repeatedly exposed whenever alcohol is consumed, the cumulative damage can gradually raise the probability that abnormal cells will emerge and begin dividing uncontrollably. In addition to damaging DNA, acetaldehyde can also form crosslinks between DNA strands and proteins. These crosslinks interfere with normal cellular processes, including gene expression and DNA replication. When these processes are disrupted repeatedly, the stability of the genome becomes compromised. Genomic instability is a key hallmark of cancer because it allows cells to accumulate multiple mutations that collectively drive uncontrolled growth. The presence of acetaldehyde in cells essentially increases the number of opportunities for these mutations to occur, particularly when exposure happens repeatedly over months or years of alcohol consumption. The formation of acetaldehyde illustrates why alcohol's cancer risk is rooted in fundamental biochemical processes rather than short-term intoxication effects. Every time alcohol is metabolized, this toxic intermediate is produced. The body works to neutralize it, but the efficiency of that process depends on both the amount of alcohol consumed and the individual's metabolic capacity. When exposure is frequent or prolonged, the balance between damage and repair can shift, allowing genetic alterations to persist and accumulate within cells. DNA damage and the resulting mutations are central to understanding how alcohol increases the risk of cancer. Inside every cell of the body is DNA, a complex molecule that carries the instructions responsible for controlling how cells grow, divide, repair themselves, and eventually die. Under normal circumstances, the integrity of this genetic information is protected by sophisticated repair systems that continuously monitor and correct errors. However, certain chemicals can interfere with this process, damaging DNA or disrupting the mechanisms that keep genetic information stable. Alcohol contributes to cancer risk largely because its metabolic byproducts and associated biochemical processes increase the likelihood of DNA damage while simultaneously weakening the systems that normally repair it. One of the primary contributors to this damage is acetaldehyde, the toxic compound produced when the body metabolizes alcohol. Acetaldehyde is highly reactive and can bind directly to DNA molecules inside cells. When this happens, it forms structures known as DNA adducts, which distort the normal shape of the DNA strand. Because the structure of DNA determines how accurately it can be copied during cell division, these distortions can lead to errors when the genetic code is replicated. When a cell attempts to divide and copy damaged DNA, it may insert incorrect genetic information into the new strand. These replication errors become permanent mutations if they are not corrected. Cells possess specialized enzymes designed to detect and repair damaged DNA before mutations become permanent. These repair systems are constantly active because DNA damage occurs naturally through normal metabolic processes and environmental exposures. However, alcohol-related compounds can interfere with the activity of these repair enzymes. When repair mechanisms become less effective, damaged DNA may persist long enough for the cell to replicate it incorrectly. Each time this occurs, the probability of harmful mutations increases. Not all mutations lead to cancer, but certain types of mutations can affect genes that control cell growth and survival. Some genes act as tumor suppressors, functioning like biological brakes that prevent cells from dividing too quickly or inappropriately. Other genes, known as oncogenes, regulate signals that stimulate cell growth and division. When mutations occur in tumor suppressor genes, the protective brakes that limit cell proliferation can become disabled. When mutations activate oncogenes, cells may receive constant signals to divide. The combination of these effects can lead to uncontrolled cellular growth, which is the defining feature of cancer. Alcohol-related DNA damage can also cause breaks in the DNA strands themselves. DNA exists as a double helix composed of two complementary strands. If one or both of these strands break, the cell must repair the break accurately in order to maintain genetic stability. However, repeated exposure to damaging compounds can overwhelm repair systems or lead to faulty repair. Incorrectly repaired breaks can rearrange sections of DNA, delete important genetic sequences, or cause chromosomes to fuse together in abnormal ways. These structural changes in the genome can disrupt normal gene function and increase the risk of malignant transformation. Another important factor is the cumulative nature of DNA damage. The body is constantly replacing old or damaged cells with new ones, and each cell division requires the entire genome to be copied. If alcohol consumption repeatedly exposes cells to DNA-damaging compounds, the number of opportunities for replication errors increases. Over time, mutations can accumulate in the same cell lineage. A single mutation might not be sufficient to trigger cancer, but multiple mutations affecting different regulatory genes can gradually shift the cell toward uncontrolled growth. Certain tissues may be particularly vulnerable to this process. Cells lining the digestive tract, including the mouth, throat, esophagus, and intestines, are frequently exposed to alcohol and its metabolites. Because these tissues experience repeated exposure and constant cellular turnover, DNA damage occurring in these cells has more opportunities to be passed along during cell division. The combination of direct exposure and frequent replication increases the likelihood that mutations will accumulate in these regions. Alcohol also indirectly increases DNA damage by creating conditions that promote genetic instability. Chronic exposure can lead to inflammation, oxidative stress, and metabolic disruptions that further challenge the cell's ability to maintain the accuracy of its genetic code. When DNA repair pathways are weakened and damaging processes are amplified, the balance between genetic damage and correction shifts in a way that allows mutations to persist. Over long periods of time, this gradual accumulation of mutations can transform normal cells into cells that grow uncontrollably and evade the body's regulatory systems. Another important mechanism through which alcohol increases the risk of cancer involves oxidative stress and chronic inflammation within the body. These processes occur at the cellular level and can gradually damage tissues, disrupt normal biological functions, and create conditions that make cancer development more likely. Oxidative stress arises when the body produces an excess of reactive oxygen species, which are unstable molecules that can interact with and damage essential cellular components such as DNA, proteins, and lipids. Alcohol metabolism significantly contributes to the production of these reactive molecules, especially when alcohol is consumed regularly or in large amounts. When ethanol is metabolized in the liver and other tissues, several biochemical reactions take place that generate reactive oxygen species as byproducts. These molecules contain unpaired electrons, making them highly reactive and capable of interacting with nearby cellular structures. Because of their instability, reactive oxygen species tend to attack molecules in order to stabilize themselves. DNA is particularly vulnerable to this type of damage. When reactive oxygen species interact with DNA, they can alter the structure of nucleotide bases, break strands of the DNA molecule, or create chemical modifications that interfere with normal genetic function. If the damage is not repaired correctly, these changes can become permanent mutations that disrupt the genes responsible for regulating cell growth and division. The body does have natural defense systems designed to neutralize reactive oxygen species. Antioxidant molecules and enzymes help stabilize these reactive compounds before they cause harm. However, frequent alcohol consumption can overwhelm these protective systems. The continuous production of reactive oxygen species during alcohol metabolism may exceed the capacity of the body's antioxidants to neutralize them. When this imbalance occurs, oxidative stress develops. Under conditions of oxidative stress, cellular components experience repeated and cumulative damage, increasing the likelihood that genetic material will be altered in ways that promote cancer. Oxidative stress also triggers inflammatory responses throughout the body. Inflammation is normally a protective mechanism used by the immune system to respond to injury or infection. When tissues are damaged, immune cells release signaling molecules that attract additional immune cells to the area, helping repair the damage and eliminate harmful substances. However, when inflammation becomes chronic, the process can begin to cause more harm than benefit. Persistent inflammation creates a biological environment where cells are repeatedly damaged and forced to regenerate, which increases the number of times cells divide. Each round of cell division presents another opportunity for errors to occur during DNA replication. Chronic inflammation also releases chemicals that can further damage DNA and surrounding tissues. Immune cells involved in inflammation produce additional reactive oxygen species and other reactive molecules as part of their defense strategy. While these substances are effective at destroying harmful pathogens, they can also harm nearby healthy cells. Over time, the repeated exposure to these damaging molecules contributes to a cycle of injury and repair within tissues. This cycle creates a setting where genetic instability becomes more likely and cells begin accumulating mutations that disrupt normal regulatory pathways. Alcohol-related inflammation is particularly evident in the liver, where most alcohol metabolism occurs. Persistent alcohol consumption can lead to conditions such as fatty liver disease and alcoholic hepatitis, both of which involve inflammation and tissue injury. As the liver attempts to repair itself, cells divide more frequently, increasing the risk that DNA damage caused by oxidative stress will be passed on during replication. If enough mutations accumulate in the same group of cells, they may begin to grow uncontrollably, potentially leading to liver cancer. Other tissues in the body can also experience inflammatory effects associated with alcohol consumption. The digestive tract, including the stomach and intestines, may develop chronic irritation due to repeated exposure to alcohol and its metabolic byproducts. This irritation can trigger inflammatory responses that weaken tissue integrity and promote the release of harmful chemical mediators. Over time, these processes can create an environment where normal cellular regulation breaks down and abnormal cells begin to proliferate. Oxidative stress and inflammation therefore act together as powerful contributors to cancer risk. The damage caused by reactive oxygen species directly alters genetic material, while chronic inflammation promotes continuous cycles of tissue injury and regeneration. These combined effects increase the likelihood that mutations will accumulate in key regulatory genes, gradually shifting cells toward uncontrolled growth and tumor formation. Alcohol can also increase the risk of cancer by influencing hormonal systems in the body, particularly those that regulate growth, reproduction, and tissue development. Hormones act as chemical messengers that travel through the bloodstream and signal cells to perform specific functions. Many tissues in the body depend on hormones to regulate when cells should grow, divide, or remain inactive. When the balance of these hormones is altered, it can change the rate at which certain cells multiply. Because cancer is closely linked to uncontrolled cell division, hormonal disruptions can play a significant role in increasing cancer risk. One of the most well-studied hormonal effects of alcohol involves estrogen. Estrogen is a hormone that plays an essential role in reproductive health and development, especially in females, but it is present in both men and women. It helps regulate the growth of tissues such as the breast and reproductive organs. However, higher levels of estrogen over long periods of time have been associated with an increased risk of several cancers, particularly breast cancer. Alcohol consumption can raise the level of circulating estrogen in the bloodstream, which may stimulate the growth of hormone-sensitive cells. When estrogen levels increase, certain cells respond by dividing more frequently. Each time a cell divides, it must replicate its DNA so that the new cell receives the correct genetic instructions. Although cells have systems to ensure that this replication occurs accurately, errors can sometimes happen. The more frequently cells divide, the greater the opportunity for these errors to occur. If a replication error affects genes responsible for controlling cell growth or DNA repair, it may create mutations that promote the development of cancer. In this way, alcohol indirectly increases the probability of harmful genetic changes by accelerating the rate of cell division in hormone-sensitive tissues. Alcohol can raise estrogen levels through several biological pathways. One pathway involves the liver, which plays a central role in metabolizing both alcohol and hormones. When alcohol is consumed, the liver prioritizes breaking down ethanol and its byproducts. This metabolic focus can reduce the liver's ability to regulate and eliminate excess hormones from the bloodstream. As a result, estrogen may remain in circulation for longer periods, increasing the exposure of tissues to this growth-stimulating signal. Over time, prolonged exposure to elevated hormone levels can contribute to abnormal cell growth. Another way alcohol influences hormonal balance is by affecting enzymes involved in hormone production. Certain enzymes that convert precursor molecules into active hormones may become more active in the presence of alcohol. This can increase the total amount of estrogen produced within the body. Additionally, alcohol can influence the function of adipose tissue or body fat, which is also involved in hormone regulation. Fat tissue contains enzymes that convert other hormones into estrogen. When alcohol alters metabolic processes within fat cells, it may contribute to increased estrogen production, further elevating hormone levels. The effects of alcohol on hormonal balance are not limited to estrogen alone. Alcohol can also influence other hormones involved in metabolic and growth-related pathways. For example, it may affect insulin and insulin-like growth factors, which regulate how cells use energy and respond to growth signals. When these hormonal pathways are disrupted, cells may receive stronger or more frequent signals to divide. Increased cell proliferation raises the likelihood that genetic errors will accumulate over time, particularly if the body's DNA repair mechanisms are already under stress from other alcohol-related effects. Hormonal disruptions can be especially significant in tissues that are naturally responsive to growth signals. In breast tissue, for example, cells contain receptors that respond directly to estrogen. When estrogen binds to these receptors, it activates genetic pathways that encourage cell growth and division. If alcohol increases estrogen levels, these growth signals may occur more often, increasing the number of times cells replicate their DNA. With each additional replication cycle, the risk of mutation increases, particularly if other damaging processes such as oxidative stress or acetaldehyde exposure are occurring simultaneously. Because hormonal systems operate throughout the body, the effects of alcohol-related hormone changes can influence multiple organs. Tissues that rely heavily on hormonal regulation may experience altered patterns of growth and regeneration when hormone levels are persistently elevated. Over time, This imbalance can contribute to an environment where abnormal cells have a greater opportunity to emerge and multiply, increasing the overall risk of cancer development in hormone-sensitive tissues. Alcohol can also increase the risk of cancer by interfering with the body's ability to absorb, store, and use essential nutrients that protect cells and maintain genetic stability. Proper nutrition plays a crucial role in supporting the biochemical processes that repair DNA, regulate cell division, and defend the body against cellular damage. Many vitamins and minerals act as cofactors in enzymatic reactions that keep the genome stable and allow damaged cells to repair themselves. When alcohol disrupts the absorption or metabolism of these nutrients, the body's protective systems become less effective, making it easier for mutations and other forms of cellular damage to accumulate. One of the most important nutrients affected by alcohol consumption is folate, also known as vitamin B9. Folate is essential for the synthesis and repair of DNA, as well as for the production of nucleotides, which are the building blocks of genetic material. During normal cell division, DNA must be copied accurately so that new cells receive the correct genetic instructions. Folate supports the chemical reactions that ensure DNA strands are formed properly and that damaged DNA can be repaired when necessary. When folate levels are insufficient, the accuracy of DNA replication decreases, and cells may incorporate incorrect genetic information during division. Alcohol interferes with folate metabolism in several ways. First, it can reduce the absorption of folate in the small intestine, preventing adequate amounts of the vitamin from entering the bloodstream. The lining of the digestive tract is responsible for transporting nutrients into circulation, but alcohol can damage these cells and impair their ability to absorb vitamins efficiently. When this process is disrupted, the body may receive less folate than it needs, even if the diet contains adequate amounts of the nutrient. In addition to impairing absorption, alcohol can also interfere with how folate is stored and transported within the body. The liver normally acts as a reservoir for many vitamins, including folate, storing them so they can be released when needed. However, alcohol places significant metabolic stress on the liver because the organ must prioritize detoxifying ethanol and its byproducts. When the liver's metabolic capacity is focused on processing alcohol, its ability to regulate and distribute nutrients may become compromised. As a result, folate levels within tissues may decrease, limiting the body's ability to support normal DNA synthesis and repair. Alcohol can also accelerate the breakdown and excretion of certain nutrients, including folate. Increased urinary excretion means that vitamins are removed from the body more quickly than usual, further reducing their availability for essential cellular processes. When these deficiencies persist over time, cells may experience repeated cycles of DNA replication without the necessary biochemical support to maintain genetic accuracy. This increases the likelihood that errors will occur in the genetic code, which can lead to mutations that contribute to cancer development. The effects of reduced folate are particularly concerning in tissues where cells divide frequently. Rapidly dividing cells require continuous production of nucleotides to build new DNA strands. If folate is lacking, these cells may attempt to replicate DNA using incomplete or faulty building blocks, resulting in structural abnormalities in the genetic material. These abnormalities can disrupt genes responsible for regulating cell growth and maintaining genomic stability. Over time, the accumulation of such errors may allow cells to bypass normal growth controls and begin dividing uncontrollably. Beyond folate, alcohol can also affect other nutrients that contribute to cellular health, including vitamins involved in antioxidant defense and metabolic regulation. Nutrients such as vitamins B6, B12, and certain antioxidants help maintain normal cellular function and support the body's ability to neutralize damaging molecules produced during metabolism. When alcohol interferes with the availability or function of these nutrients, the body's capacity to protect and repair cells diminishes. This combination of reduced nutrient support and increased exposure to damaging compounds creates conditions where genetic instability becomes more likely. Because nutrient deficiencies may develop gradually over time, individuals who consume alcohol regularly may not immediately notice their effects. However, even moderate interference with nutrient metabolism can have long-term consequences for cellular health. When the systems responsible for maintaining DNA integrity are weakened, the balance between damage and repair shifts. As cells accumulate genetic alterations without sufficient repair mechanisms, the probability that some of these cells will develop cancer-promoting mutations increases. Alcohol can also increase the risk of cancer by weakening the body's immune defenses and making tissues more vulnerable to damage from carcinogens. The immune system plays a critical role in maintaining health by identifying and eliminating abnormal cells before they have the chance to grow into tumors. Every day, cells in the body experience small genetic changes as a result of normal metabolic processes or environmental exposures. In most cases, immune cells are able to detect these abnormal cells and destroy them before they can multiply. This process, often referred to as immune surveillance, acts as an important protective mechanism against cancer. When alcohol interferes with immune function, this protective system becomes less effective, allowing damaged or mutated cells to survive and potentially develop into cancerous growths. Chronic alcohol consumption can impair several components of the immune system. Immune cells such as T cells, natural killer cells, and macrophages play key roles in identifying and eliminating abnormal cells. Alcohol can reduce the activity and efficiency of these immune cells, weakening their ability to detect cells that have undergone genetic mutations. When immune surveillance is reduced, abnormal cells have a greater opportunity to continue dividing and accumulating additional mutations. Over time, this can allow clusters of abnormal cells to grow into tumors that might otherwise have been eliminated by a fully functioning immune response. In addition to weakening immune surveillance, alcohol can also disrupt communication between immune cells. The immune system relies on signaling molecules called cytokines to coordinate responses to infection, inflammation, and cellular damage. Alcohol can interfere with the production and regulation of these signaling molecules, altering how immune cells communicate with each other. When communication becomes less efficient, immune responses may become slower or less targeted, further reducing the body's ability to control the early stages of cancer development. Alcohol also increases the vulnerability of tissues that are directly exposed to it, particularly those in the mouth, throat, and digestive tract. When alcohol is consumed, it comes into direct contact with the cells lining these areas before it is absorbed into the bloodstream. Alcohol acts as a chemical solvent, meaning it can weaken and disrupt cell membranes. Cell membranes normally act as protective barriers that regulate what enters and leaves the cell. When these membranes are compromised, harmful substances from the environment can enter cells more easily. This increased permeability becomes particularly important when other carcinogens are present. For example, tobacco smoke contains many chemicals that are known to damage DNA. When alcohol weakens the protective barriers of cells in the mouth or throat, it becomes easier for these carcinogens to penetrate the cells and interact with genetic material. This interaction significantly increases the likelihood of mutations forming within the DNA. This is one reason why individuals who both smoke and drink alcohol have a much higher risk of developing cancers of the oral cavity, throat, and esophagus compared with those who engage in only one of these behaviors. Alcohol can also irritate and inflame tissues in the digestive tract. Repeated exposure to alcohol can damage the lining of the stomach and intestines, leading to inflammation and increased cellular turnover as the body attempts to repair injured tissue. When cells divide more frequently during the repair process, the chances of errors occurring during DNA replication increase. Combined with the weakened immune surveillance caused by alcohol, these damaged cells may persist instead of being removed by the immune system. The combined effects of tissue vulnerability and weakened immune protection create conditions that make it easier for carcinogenic substances to cause lasting cellular damage. As abnormal cells accumulate genetic mutations and avoid elimination by the immune system, they gain more opportunities to multiply and form clusters of cells that grow beyond normal biological controls. Over time, these processes can gradually transform healthy tissue into environments where cancer development becomes more likely. When these effects are viewed together, it becomes clear that alcohol's influence on cancer risk is not driven by a single pathway, but by the interaction of multiple biological disruptions. Weakened immune surveillance allows abnormal cells to survive longer than they normally would, while increased tissue vulnerability allows carcinogens to penetrate cells more easily and damage DNA. At the same time, repeated irritation and inflammation force tissues to repair themselves more frequently, increasing the number of times cells must replicate their genetic material. Each of these factors raises the probability that harmful mutations will accumulate. As this process continues over months and years of exposure, the body's natural defenses against abnormal cell growth gradually become less effective, allowing damaged cells to persist, multiply, and eventually develop into cancerous tumors.